Hello everyone, welcome back to my channel Competitive Botany. In this video also we are going to see the remaining questions of APSET Life Sciences previous paper 2021. Let's see the questions one by one. First question, staining with fluorochrome is involved in. Options are G banding, O banding, Q banding, C banding. The correct option is third one, that is Q banding. In cell cycle transcription of RNAs and translation into proteins occurs during. Options are G1 and S, G2 and S, G1 and G2, G1, G2 and S. Correct option is third one, that is G1 and G2. The proteinaceous molecular glue which holds sister chromatids together during chromosome separation at Anaphase is. Options are cyclosome, cohesion, separate, securing. Correct option is cohesion. One of the following techniques is useful for testing fluid samples for microbiological contamination. Options are viable count, spectrophotometer, electronic Coulter counter, membrane filter count. Correct option is fourth one, membrane filter count. Next question. In this type of culture, all the cells of the particular population are in the same stage of development and divides simultaneously. Options are enrichment culture, batch culture, continuous culture, synchronous culture. Correct option is synchronous culture. Which of the following pairs is not correctly matched? Options are in Casuarina, the pollen tube enters into the ovule through chalaza. Oenothera follows tetrasporic type of embryo sac development. In Fritillaria, a three-celled egg apparatus is present. In Asaphodilis tenuifolius, the endosperm development of helobial type. Correct option is Oenothera follows tetrasporic type of embryo sac. Onagra type of embryogeny is present in. Here there are 4 examples given. So from these we have to pick the 3 correct examples. They are example for Onagra type of embryogeny. Okay. So the correct option is, first one is the correct option. That means Arabidopsis thaliana, Capsella bursa pastoris, Ceratocephalus foliatus. They contain Onagra type of embryogeny. Next question. Protoderm, procambium and ground meristem are classified on the basis of. Options are position and occurrence, characteristics of initial cells, functions, cell division and growth pattern. Correct option is functions. Phloem furrows are formed in the secondary stem of. Options are Boerhaavia, Acheranthus, Dracaena, Bignonia. Correct option is Bignonia. Next question. Here we have to match the phyllotaxy types. Okay. So let's see the matching directly. So leaves closely overlapping one another. So the type of phyllotaxy is called as imbricated. Leaves in 2 rows with the third leaf above the first leaf, that is referred as distichous. More than 3 leaves at each node, that phyllotaxy is called as whorled phyllotaxy. Bearing pairs of leaves at each node and the pairs of successive leaves at right angles, the type of phyllotaxy is called as opposite decussate. Next question. When 2 authors jointly publish the name of a plant, the names of both authors are connected by. Correct options are Ex-Situ et al., at or end. The correct option is at or end. So here also we have to match the list 1 with the list 2. I am going to give you the matching directly. Sporocarp present in Marsilia. Trabeculae in stem seen in Salacinella. Antheridia 4 and Archegonia 4 are present in Markantia. Vallecular and carinal canals in stem present in Equisitum. Next, here also the matching is given. We have to match the list 1 with list 2. Here the plant diseases are given. So I am going to give you matching directly. Citrus canker caused by Xanthomonas and Axanopodis. Green ear of Basra caused by Sclerospora graminicola. Red rot of sugarcane caused by Colitotrichum falcatum. Late blight of potato caused by Phytophthora infestans. Next question. One of the following is an example for Ex-Situ conservation. Options are national parks, botanical gardens, biosphere reserves, sanctuaries. Correct answer is botanical gardens. Next question. The function of primosome is, options are initiation of Okazaki fragments on the lagging strand, initiation of Okazaki fragments on the leading strand, initiation of Okazaki fragments on both leading and lagging strand, joining of Okazaki fragments. Correct option is the first one, that is initiation of Okazaki fragments on the lagging strand. Next question. The enzyme component of E. coli's reprosome is, options are DNA primase, DNA polymerase 1, DNA polymerase 2, DNA helicase. So here they have considered two questions as the correct options. They are DNA primase and DNA helicase. Both are correct for this question. Match the components of RNA polymerase holoenzyme list 1 with their respective functions list 2. Here they have given holoenzyme subunits. So we have to match them with their respective functions. Okay, I am going to give you matching. Alpha subunit involved in the assembly of tetrameric core. Beta subunit contains ribonucleotide triphosphate binding site. Beta prime subunit contains DNA template binding site. Sigma subunit contains initiation of transcription. Sigma subunit involved in the initiation of transcription. Next question. So the sequence complementary to Shine Dalgarno sequence is located on, options are 5S rRNA, 5.8S rRNA, 16S rRNA, 28S rRNA. Correct option is 16S rRNA. Next question. The base inosine found in several tRNAs is the post-transcriptional modification of, options are adenosine, cytidine, guanosine, thymidine. Correct option is adenosine. Next question. A folded region of riboswitch capable of binding a specific metabolite is called as, options are aptamer domain, expression domain, amino acid binding domain, or loop. Correct answer is aptamer domain. Next question. When the expression of genes is controlled by its parental origin, the phenomenon described as, options are gene attenuation, gene imprinting, chromatin remodeling, feedback inhibition. The correct option is gene imprinting. Next question. Read the following statements in the context of lac operon in E. coli. So first generalization is lac Z, lac Y, Lac A produce different enzymes. Second one is Lac O is the site for repressor binding protein and codes for protein. Lac P site for RNA polymerase binding and codes for a protein. Lac 1 produces repressor protein. Among these we have to pick the correct statements. So answer is third one. First and fourth generalizations or first and fourth statements are correct regarding the lac operon in E. coli. Next question. Base excision repair is initiated by the enzyme. Options are S1 nuclease, DNA exonuclease, restriction endonuclease, DNA glycosylate. Correct option is DNA glycosylate. Next question. Match the following. Here also we have to match these chemical mutagens with the correct examples. I'll give you matching directly. Alkylating agent. Example mustard gas. Base analog. Example 6-bromouracil. Deaminating agent. Example nitrous acid. Ecridin agent. Example proflavin. Next question. A cytosine in the DNA sequence of a gene is substituted by thymine. This type of base change is an example of. Options are tautomerism, frame shift mutation, transversion and transition. Correct option is transition.